Hello, and thank you for joining us. Today's webinar is Single Cell Analysis with the MVX 7100 Workstation, presented by Dr. Pete Winship of Teledyne CETAC. This webinar is being recorded and will be made available for viewing within a couple of days. You will receive an email notification of its availability. Okay, now I'll turn it over to our presenter, Dr. Pete Winship.
Hello there. Hi. Shelley, thank you for the kind introduction. So my name is Pete Winship. I'm a product manager at Teledyne CETAC Technologies. I'm responsible for the MVX 7100 microliter workstation. That's one of the products I look after. So today I'm going to give a presentation on the use of this for single cell analysis. So I'll start off with just a quick agenda. So just to go through what I'm going to walk through. I'll give a brief introduction to cell analysis generally, which will take in some of that ICP-MS, and particularly with focus on cells. I'll be talking for the most part on the idea of a workflow about starting with cells, going through to data, and I'll give some examples of some data that that's been published recently with one of our collaborators over at the University of so that's what I'm going to go through today. So I'll start off with the introduction to cell analysis with ICP-MS. I've been involved in ICP-MS for around about 20 years now, as have a number of our colleagues, and in that time period, a lot has happened in trace element analysis, and particularly with ICP-MS instrumentation. So ICP-MS instrumentation today is considerably more sensitive, it's more robust, it's faster, the instruments are smaller, and it's capable of so much more than it was 10, 20 years ago. So as part of this development, it's now possible to capture data from what can be referred to as single events. So we're talking about data that can be captured from small components of a liquid sample. So, for example, particulate material that might be, excuse me, in the micro or the nanoscale, and for what we're going to talk about today, biological cells, which are in the low end micrometer scale. So cells generally are a huge area of research in biological and clinical applications. They are studied these days for elemental composition, as well as all the other aspects of analysis that they're part of. And excuse me, so that cells are studied as an entity themselves, or as part of a study or a project, as a means to an end, essentially looking at cells as part of a wider study. So we could be looking at or interested in the elemental content of individual cells. We could be interested in cellular uptake, so we might be interested in species which are of a nutrient origin. We might be interested in species in cells which are present from a contaminant point of view, or we might be interested in looking at cells which are involved in metal-based drug uptake, so compound uptake studies, or nanoparticle uptake. So because ICP-MS can now capture data for this kind of sample, it's now been brought into broader study on biological cells. Excuse me. So how does this work? So typically, if we ignore solid sample analysis with something like laser ablation, a majority of sample analysis in ICP-MS is undertaken with liquid sample introduction. So what you typically have is a homogeneous sample or close to a homogeneous liquid sample, and that sample is introduced to a plasma, an ICP, in an instrument via a nebulizer and spray chamber with some sort of automation. So this sample is introduced to the ICP, and on the image here that we have on the top left-hand side of the screen, we've got a plasma torch here, and you'll see these green dots going through representing homogeneous sample going through the plasma for atomization, breakdown, atomization, ionization, and detection. So the kind of signal that we see from this kind of sample going into ICP-MS is represented in the plot on the top right here, where we have a consistent signal coming through, a stable signal from the continuous introduction of the liquid sample. So that's represented by the green line there. Now, in something like cell analysis, we don't have that. In cell analysis, we have we're looking at probably a suspension of cells in a liquid sample, and those cells are introduced in the same way through a nebulizer and spray chamber to an ICP, typically with some sort of automation on the front end again. But what we have here, instead of this homogeneous sample where we get a continuous introduction and a continuous signal, what we have instead is that as liquid is introduced to a nebulizer, we have cells coming through from the suspension, and these cells will hit the plasma. A cell will be broken down, atomized and ionized in the same way, but instead of that continuous signal from that continuous introduction, you get an ion plume. So more like an individual event, so like a pocket of ions going through the system. Therefore, what you see in terms of a signal is this plot that's on the right here, so this time versus intensity plot, where we get a spike. So as the cell passes through the plasma, we get breakdown, we get ionization of the analytes of interest to us, and that is the analytes are then seen as a spike as we go through. Now, if the dwell time is set correctly on an ICP-MS instrument, we can capture that spike as it comes through, and we can work with that data, we can process that data, and we can get the data that we want from cells as they pass through. So this is pretty much what I've described here. The cells going through the plasma, forming ion plumes that we can capture. Now these events, these spikes as they go through, these events, the number of these events is equivalent to the number of cells as it goes through the plasma. So the cell concentration per unit volume. The intensity of these events is equivalent to the element quantity. So much like we're doing detection on a homogeneous sample, we are able to take this data and turn it into concentration data or mass per cell data, for example. So what we see here is that the kind of plot that we will typically see down here on the bottom right of the slide here. This is a time versus signal intensity plot here, and what you're looking at here is these individual spikes that are coming through. These spikes represent cells which have passed through a plasma, been ionized, atomized, and detected. And these spikes raised above the background are measurable with the ICP-MS operating software. So that's the process in a nutshell, what we're looking to do. And this is the same process as you might be interested in microparticles or nanoparticles. It's the same process, the same kind of approach to capturing data. So that's generally the process. So, but there's more to this. There has to be more to this. It's not that simple, unfortunately. So what I'm going to do next is I'm going to just describe the workflow of this process. What's involved in generating and capturing this data? What are the variables that we need to be aware of? So what I've put down here is an analytical workflow. starting with so a flow of what happens through the process of sample introduction. Now this won't look particularly unusual to most people who operate ICP-MS. We start off with a sample in some form of sample position. That sample will then be carried through some sort of automation, through some automation components, tubing, a pump, etc. The sample then is carried through some form of interface glassware, so which is typically a nebulizer and a spray chamber. So a nebulizer to generate fine droplets from the liquid sample, and then a spray chamber to filter those droplets according to size, so that you get a nice stable signal in ICP-MS in your instrument, and then you capture your data. So that's typically what's going on here. But the thing to consider from the very outset of this is cells. Cells are a different thing entirely to an ICP-MS instrument, and the key to success here is essentially making sure that you maintain cell integrity from A to B. So you maintain the integrity of the cells starting at the sample position through to the ICP-MS. So you want what they refer to as the transport efficiency, the maximum transport efficiency. You want to maintain the cell integrity for as many of those cells from one side to the other. So which is where the key to this lies. And there's a number of variables associated with this. So I'm just going to work through these individual stages, starting with the idea of where we start with the sample position, what might be involved there. I'll go on to automation components, but this is going to be focused on the MVX 7100 system, which is what we use for single cell work, and I'll go into why we do that shortly. Then I'll move on to the idea of glassware, and then the kind of variables associated with the ICP-MS detection, what the key things are there. So that's our workflow. So starting with the sample position, so there's a number of things we need to consider here, because we're talking about cells that we want to be detecting. It's not as straightforward as just having cells racked up in sample tubes that you might have in some sort of water analysis. So what we have to consider here is that your cells might have been grown up in cell culture. They might be a certain type of cell, certain size, a certain shape. They might be held in some sort of media. There might be conditions to maintain, for example, temperature. You might have to consider things like whether a sample will settle, cells will settle according to gravity perhaps over time. So there's a number of things to consider with your sample. There's also things like the concentration of the cells in your sample, because if you have too many cells in your samples, you introduce it to your ICP-MS, you're going to overwhelm the actual measurement side of things, and it might be less easy to detect the data that you want out of this. So in the same way that you would introduce your samples to ICP-MS, you have to start somewhere with some form of receptacle for your samples. So that may be well plates, so say a 96 well plate or a 384 well plate, but you could also work with sample tubes and vials, for example. So these are considerations that you have to factor into the kind of work that you're doing here with the ICP-MS, which is where the MVX 7100 comes in, because we've opened that up to a lot of these variables, and it's very flexible in terms of this analysis. So the automation. So we start with our samples in well plates or tubes in a certain media. And what we have here is for our sample introduction to ICP-MS for single cells, this is what we've been working with, the MVX 7100 system. So this is a syringe-driven autosampler as a front end for ICP-MS, which is more appropriate for single cells. Typically for ICP-MS, you would use a peristaltic pump where you are using the peristaltic process to introduce some liquid samples through tubing to a nebulizer. That is not as appropriate for this kind of work, unfortunately. The cells aren't going to respond very well to that kind of pressure, that kind of pressing that goes through the pumping process. So a syringe-driven approach is more appropriate here. So this system will allow you to take up samples of lower volumes down to around about 5 microliters and up to about 1.5 milliliters. That's the standard, but it is flexible beyond there. The system will also allow you to very, very accurate control over the sample uptake flow rates and the introduction flow rates. So what we have here on this system is we have 2 syringe pumps. You'll see in the image there's one on the right-hand side and one on the left. The one on the right-hand side of the image is responsible for pulling the sample up onto the sample loop. And we can do that as fast or as slow as we like to make sure that we don't introduce that any particular agitation to the cells that might lead to lysis. And then once we pull the sample up via that pump onto a sample loop on a 6-port switching valve, the other syringe pump takes over and introduces that sample to your nebulizer and spray chamber and then the ICP-MS. And that flow rate, again, is well controlled, and that can go all the way down to 5 microliters and then all the way up to and well beyond any introduction flow rate that's required for any nebulizer that's available on the market. The system can also provide a degree of sample homogenization. So if there are issues with larger cell sizes and any kind of settling in a well plate or in a sample vial, a degree of homogenization can be introduced to this to try and redistribute the cells in the suspension again before introduction. There's an option to have temperature control on this as well. So you can go down to 4 degrees centigrade or up to 40 degrees C. So if you need cooling, it's there. If you need to hold your samples at more of an incubation temperature, say around about 38 degrees C, that's available to you as well. And then we can have either sampling from stepped and piercing vials, tubes, or 96 or 384 well plates, depending on what you want to work with for your cells. So we start there with the automation. But between the automation and the ICP-MS, we need the right kind of glassware. And given what we're working with here, we're taking cells and we're introducing them to ICP-MS, and the key is keeping them intact, making sure that they don't split, they don't lyse in that process, then you need the right kind of glassware between the two. So we've been working with Glass Expansion over in Australia, who've developed this glassware interface for this application with individual cells. So this is a nebulizer and spray chamber combination for cell work. So this is a high efficiency nebulizer which has a nebulizer which is set up for introduction flow rates at less than 50 microliters per minute, though you don't need to go as high as 50. The introduction flow rate is around about 10 to 20, that kind of window. The internal capillary of the nebulizer is quite a wide bore. The intention there is to make sure that your cells have got some room to move, that you're not forcing your cells together prior to reaching the tip of the nebulizer for the nebulization process. Again, you're trying to avoid cells lysing, splitting in this process. So you're trying to make sure that they maintain that integrity through the process. And then nebulized droplets are then introduced into this spray chamber. So this is a high efficiency spray chamber with additional gas flows to guide the nebulized droplets to the ICP-MS. Now this is because we're looking at flow rates around about 10 to 20 microliters per minute. And we're talking about typically low volumes, then this is nearly a total consumption process. So total consumption nebulizers run a bit slower than this, but it's not that far away. So this is a near total consumption process. So the majority of your nebulized liquid, your droplets in your cells, the majority of that is going into ICP-MS. So, and then the next slide is this. So just show that's what the glassware looks like with the nebulizer going into the back of the spray chamber here, and there's an additional port. You can just see that on the spray chamber at the peak end at the top there. And this spray chamber attaches directly to your torch, and that's how you get your introduction from your MVX 7100 through the glassware to your ICP-MS. So how do we know that the cells maintain that integrity? Well, we know from 2 points of view. We've done a fair amount of testing on this. So firstly, we know from the data that is generated from the ICP-MS and the peaks that we get, the events that we detect through this sample introduction. So we know, we see it in the data, but also we've studied the cells in terms of microscopy at each step as they go through the automation and then through the nebulization and separation in the spray chamber process. So there's some images here for you just to see. So this is an image from a leukemia cell line. So these are cells which are 10 to 20 micrometers in their largest dimension. Now these cells are widely used in research and they're easy to culture and isolate. So these are cells which have been selected for this purpose. So the images that were shown here, this is work that has been undertaken through a collaboration with Agilent Technologies and through one of our collaborators at Chiba University in Tokyo. So this is where these images have come from. But these are leukemia cells. What you'll see here is that if we start on through this sort of flow here on the left-hand side, what you see here are the furthest left image is these are these cells in a truss buffer. And that's what the cells look like before anything's been done to them. Those cells were then introduced to, or were taken up on our MVX 7100 microliter workstation system. And the cells have been taken through tubing, they've been taken through a 6-port valve, they've been taken up via a syringe pump. And we know that those cells have maintained integrity through to the point of view where they hit a nebulizer, where they're introduced to a nebulizer. Now, we know that these cells survive the nebulization process. They survive going through the spray chamber and into the ICP-MS when the conditions are correct with nebulizer flows and sheath gas flows, for example. So what you'll see here is that on the right-hand side, in the within the jagged line, the broken line box there, you've got the image on the top there at a nebulization gas flow of 0.1 liters per minute. You'll see that the cells remain intact as they go through into the ICP-MS. But if the conditions are incorrect, so say if the nebulization gas flow is too high, so say if you ramp it up to 0.7 liters per minute, you'll see that some of the cells remain intact, but some of the other cells will split and live. That's what you'll see there on the bottom right-hand side there, an image of a live cell. So what we need is these cells need to maintain this integrity through this whole process. If they do live, then what happens is that the analytes that you're interested in, all that data that you want to collect, will basically be lost into the background of the measurements. So they need to stay intact so you can realize that peak as it comes out and is detected by the ICP-MS. But we know from microscopy, this has been proven by microscopy, by these images, and through the data that we see through measurements. So the ICP-MS side. So there's, like I said, over the past 20 years, there's been a huge amount of development, and that's just in the time that I've been involved in ICP-MS. It's been going on long before that. but the more sensitivity that we can get for this measurement with the ICP-MS, the better. The more accurate the data is going to be. The analyte suite, the analytes of interest, this is another area of a lot of discussion at the moment. So there's two approaches to this kind of work, and typically it's either being done by quadrupole-based ICP-MS, so either with a standard quadrupole ICP-MS or a triple quad ICP-MS, or by one of the new time-of-flight ICP-MS instruments. So you've got to then start questioning, what kind of data do I want to get from this? So am I looking for multiple analytes, which I would recommend? So taking your analytes of interest, which might be something like platinum, for example, but also with cofactors as well. So some sort of comparative element of interest, so say phosphorus, for example. So your analysis on the cells that are passing through might be multiple analytes. In the case of time of flight, you're able to capture more data, like and a full suite of a full analyte suite through the time of flight instrument. But it's possible with the most recent generation of ICP-MS to capture all of this data. And then once you've got that data, what do you do with it? So there's a number of types of data that you can take from this. You can take, much like you're doing with standard ICP-MS, you can convert the data into concentration data. That could be concentration of analyte per population of cells in your sample, or it could be something like the mass of analyte per unit, whatever it is you're interested in. So the mass of an analyte per cell, the mass of an analyte per population. It depends on the kind of data that you want to generate from this. So there's a lot that can be taken out of this process, this analysis. So an example for you. So last month, one of our collaborators at the University of Oviedo published an article in Current Trends in Mass Spectrometry on this automated process using the MVX with the MVX 7100 system with this glassware with an ICP-MS for single cell work. Now they focused on a number of different types of cells, and they looked at phosphorus, selenium, and chromium measurement through this process. So I'm just going to sort of work through some of the highlights of what they put into the article, but the article is available online if anybody wants to have a look at this. So the instrumentation that they used for this, they used a combination of the Thermo ICAP triple quad ICP-MS with our MVX 7100 microliter workstation and this glassware from Glass Expansion. So they had they did some development on this. They found the most appropriate gas flows for the cells they were working with. So that's the nebulizer gas flow and the sheath gas flow. They were introducing the sample volumes at 10 microliters per minute, which is particularly slow. This is a very slow introduction flow rate, and certainly not something that is particularly easy at all to achieve with a peristaltic pump, but is very easy to achieve with a syringe pump driven system like the MVX 7100 system. So they were introducing these samples at that flow rate with a dwell time of 5 milliseconds. So they were able to capture those single events as they went through. Now, they were looking at phosphorus, selenium, and chromium, and they were using, in this case, to avoid polyatomic ion interference, using an oxygen cell gas to oxidize the analytes of interest, and therefore they were measuring phosphorus at 47, they were measuring selenium at 96, and they were measuring chromium at 68. Now, as part of the work that they did, they've looked at a number of different cell types, and what is what should be remembered through this kind of work is that cells are very different. by their nature. Obviously, there's of all the cells, that all the mammalian cell types that people are interested in, they are all they all have different degrees of robustness. Therefore, their transport efficiency between them is going to be different. So there is no one-size-fits-all set of conditions for the cells that you might be interested in. So this is another part of the optimization of your system. So what the authors of this article have done is that they looked at some transport efficiency between the cells that they were interested in, and that they've first of all confirmed population sizes by flow cytometry, and then they've looked at different cells and the transport efficiency that they get through the same system at the best conditions. So they have looked at some human ovarian carcinoma cells, which with a cell size of approximately 8 micrometers in the largest dimension. And for those cells, they were finding transport efficiency of around about 50 to 58%. So that's 50 to 58% of the cells that were in their sample position, their sample vial or the sample tube or the well plate. 50 to 58% of those cells were surviving the process of uptake and introduction through the nebulizer and spray chamber into the ICP-MS. They did the same work for some fibroblast cells, which were a little bit larger, about 10 to 15 micrometers in the largest dimension. And they were getting they certainly would appear to be a bit more robust, but for the same number of samples per unit volume, number of cells per unit volume, those fibroblast cells were surviving this process to a greater degree. So the transport efficiency was between 85 and 95%. That's 85 to 95% of the cells surviving the introduction process. And then they looked at some yeast cells as well, with a transport efficiency of 62 to 69%. The introduction of different kinds of yeast cells here. So this is time-resolved analysis detection of phosphorus and sulfur at a mass discharge of 47 and 96. So this is yeasts where this has been confirmed by flow cytometry. You have 50,000 cells per milliliter. Now these are being introduced through the MVX and the Glassware. So for regular yeast, which is the two graphs on the left-hand side there, you've got phosphorus detection on the top and sulfur underneath. Sorry, selenium underneath. So what you've got there is for the phosphorus, you'll see these individual events, these individual spikes over time in seconds there against the intensity. And you'll see these spikes, these represent individual cells as they've been introduced to the ICP-MS. Now, you don't see that below for the selenium. So the selenium, the concentration of selenium in these cells isn't high enough to be raised above the background. But for a selenized yeast, what you'll see here is that, again, you've got the same individual events for phosphorus, as that's being introduced. But what you'll see here is that you've got, for the selenium, you've got spikes again, these individual events. So what you'll see here is that these individual events, again, like the phosphorus, represent individual cells being introduced to your ICP-MS, and that is selenium inside the cells. Now, what I've not included on this slide is that there's actually more data to support this. So if you take the regular yeast, and if you just spike the liquid sample with selenium, then you will get the same graph that's under the regular yeast here. You won't get the same as the spikes from the selenized yeast, because that is just selenium, which would appear as background. It's not going to give you these individual events. So this shows that we're successfully introducing individual yeast samples, and we're capturing that data for processing later on. So the same can be said for phosphorus and chromium data. So in terms of different yeasts, you can get yeast spiked with selenium or chromium from a nutritional point of view. And again, we're seeing here time-resolved analysis detection of phosphorus and chromium at master charge 47 for phosphorus and master charge 68 for chromium. Like the last set of yeast samples, this was measurement of 50,000 cells per milliliter, and we're successfully capturing data here for phosphorus. These are what these individual spikes are, these individual events, for both phosphorus and for chromium as well. And then there's something similar as well for the next slide, which is phosphorus in these fibroblast cells. So this is just phosphorus data here, again, individual events, but this was for 25,000 cells per milliliter here, which is confirmed by flow cytometry. So, as I mentioned before with the ICP-MS, this data, this is just time-resolved data, but this data, as it's captured, is then converted into the data that you want. So whether that is mass of an analyte per cell, whether it's mass of an analyte per population of cells, this capability in ICP-MS for individual cells allows analytes of interest in cell populations, and it's all about maintaining that integrity of cells as they're introduced into the system. So I'm going to leave that there for the day from the point of view of this data and this presentation. But I'd like to acknowledge some of our collaborators here. There's a huge number of people from Agilent Technologies that we've been working with and collaborating with over the years. So I've just put down Agilent Technologies for an acknowledgement because there's so many people involved. Glenn Russell from Glass Expansion, who we've been working with on the Glassware, Jörg Bettner and his group at the University of Oviedo in Spain, and Dr. Tanaka and Professor Ogra at Chiba University in Tokyo in Japan. So thank you very much to everybody there, and thank you for your attention as well. I imagine that there'll be a number of questions as to this kind of work. Probably the best thing to do is if you could send them to me via email, and then I can address them individually, because some of these questions might be quite broad, quite large questions to answer. So please feel free to reach out to me at the email address below, and I will come back to you with and we can discuss any questions that you have. In addition to that, we have a number of webinars in the schedule for 2020 so far, and there's a number more webinars being scheduled after these as well. So we've got a couple more tomorrow, and then we've got more through May and June. So if any of these are of interest, please drop by. Thank you.